Okay, our rising star in this session is Dr. Charles Danko, well known as a local and member of the Baker Institute for Animal Health and member of the faculty in the Department of Biomedical Sciences. So Charles received his PhD in bioinformatics from SUNY Upstate, and then he came to Cornell to do postdoc training with Adam Siepel and Lee Krause, and also working closely with John Liss here. His work combines computational biology with molecular biology approaches to really understand the intricate molecular mechanisms that control gene expression. And his lab has played a lead role in developing some key new technologies like ChRO-seq, along with important new software packages and data analysis pipelines. And he's applied these technologies to a number of interesting questions of relevance to cancer and immune function and evolution. So Charles recently completed a stint as the Robert N. Noyce Assistant Professor in Life Science and Technology, and he finished that because he was recently promoted to Associate Professor with tenure here at Cornell. He's a great choice, along with all the other outstanding speakers today, for this lineup as a rising star, and I'm looking forward to hearing about his work on transcription as a lens for interpreting genome sequence. Thank you, Bob.
Thanks. Can you hear me? Can you see my slides? Yeah, thanks, Bob, for that great introduction. Thanks to Praveen for organizing this. It's a lot of fun to be here today and talking to you. So I'm going to start this talk, as I start any talk, with the central dogma of molecular biology, which is the core idea that DNA sequence is converted into mRNA by a process known as transcription. And my lab, I give them the broad and perhaps too broad charge to understand how the DNA sequence itself controls the timing and abundance of mRNA that's produced in cells. And I think that this is a really important problem because the amount of mRNA that a cell produces is one of the key factors that determines this complex cellular behavior at the cellular level that then goes on to control phenotypic variation like size of an organism or coat color or whatever, as well as within the lifespan of an organism, changes to cellular behavior that can lead to diseases like cancer, like this mast cell tumor in a yellow lab. You can tell I'm at a vet school. So that's really our charge. And gene expression, the amount of mRNA, is controlled at the chromatin level by regions known as transcriptional regulatory elements. So if this region represents chromatin, where these represent histones and the intervening gray represents DNA, this transcriptional regulatory element is carved out when a transcription factor, represented by this green protein here, binds to DNA by recognizing a DNA sequence that it binds to. And then once it's bound, it recruits a variety of different transcriptional coactivators, which decorate the chromatin with a variety of post-translational modifications, as well as recruiting the RNA polymerase complex. So this basic process occurs at both promoters and enhancers, and then at a subset of these regulatory elements that we call promoters, the RNA polymerase is stable for a megabase in some cases, and transcribes just chugging along on the DNA to produce a processed mRNA. So really, I think it's the transcription factor, the way that I think of this, that kicks this whole process off. And so I think a really fundamental thing that we'd like to be able to do if we're going to understand a cancer genome or whatever is to be able to identify the location of these transcription factor binding sites. Where do they bind, how much, and how do they regulate gene expression? Of course, the challenge with this is that these transcription factors bind to very weak DNA sequence content, right? So here's an example of an ELF1 binding site, and you can see that it's short, only about 12 base pairs, and it's really highly degenerate, which means that there's a lot of different variation in the sequences that actually bind. So there's only about 6 base pairs of information content. It's hidden among 3 billion nucleotides in a mammalian genome. And so you really are looking for a needle in the haystack, right? You need to be able to whittle down what you're actually looking at to understand what is relevant and what is not. And the way that we do this as a community is we collect more information about genome function through these functional genomic assays and use these to identify which regions are controlling gene expression and which are not. So the assay that is the choice of my lab in a lot of cases, and this is almost exclusively what this talk is about today, is an assay that measures RNA polymerase developed by John Liss, who's here in the audience, as well as Ho-Jun Kwak when he was in John's lab as a graduate student. And this assay essentially looks for RNA polymerases. So here the height of the histogram represents the quantity of RNA polymerase at each position in a genome, and the blue and the red tracks represent the orientation of that RNA polymerase, whether it's on the Watson or the Franklin strand. And the reason that I like this assay so much, and a lot of the argument that I'm going to give you today, is that RNA polymerase is a really rich picture of what our genomes are actually doing. So you can see that through the shape of the signal at these regions, you can identify the location of enhancers, you can identify promoters, gene bodies, and even the polyadenylation cleavage site. You can find the boundaries of these transcription units. And in general, if we had the computational tools to really refine this rich picture of what our genomes are doing, we could use this assay as a strategy to maximize the information that we're gaining about what the genome is doing in a limited sample like a cancer genome. Okay? So looking at this in another way, RNA polymerase is situated in the central dogma right between these other two really privileged stages at which gene expression is controlled. So it's right between the chromatin stage and the mRNA stage, and it contains features of both the chromatin and the mRNA regulation. So we've known for decades that RNA polymerase is a very good, correlates very well with the amount of mRNA that is produced. So just if you integrate the signal in the gene body here, the correlation with mRNA-seq data is really very strong, okay? So we know that. And one of the things I really wanted to do when I started my lab is to take this kind of information in PRO-seq data and decompose it into these different histone modifications, this regulatory layer of information, and ask if we could sort of take the single assay and decompose it into a lot of different ChRO-seq tracks. So that's really our objective, is to develop a strategy, a single assay that we can use to maximize the information that we gain about a limited sample. So there's a few things that we'd like to be able to do in this kind of a context. So first, we'd like to be able to measure gene expression, and I've shown you that we can do that with PRO-seq. Second, we'd like to really be able to assay this regulatory layer, right? We'd like to be able to look at the epigenomic information, the activity of promoters and enhancers, and I hope you'll forgive the use of the word epigenomic. I use it here in the context of these histone modifications. That's what I refer to in this talk. And then we would like to be able to do both of these things at single cell resolution. So today, I'm really going to focus on these 2 points here. And then I'll show you a brief vignette where we use these tools to study the regulatory activity in a cohort of glioblastomas, so human brain cancer. So the first goal is to extract the regulatory information from ChRO-seq data. So essentially what we're doing is we're taking RNA polymerase tracks and we're converting it into tracks of chromatin state, okay? So it turns out that most of the information that we need to collect from the PRO-seq data to do this kind of conversion is the shape of the RNA polymerase, right? This is in essence an image recognition problem. Now, image recognition has in computer science circles, this is thought of as one of the harder problems that you can take on. So for those of you who read XKCD religiously, like me and graduate students that I've had in the past, there have been cartoons about this, right? So in this particular case, they're joking about being able to identify a bird in a picture, and it takes a research team in 5 years. I'm going to argue that we have solved this particular problem, but it really has taken a research team in 5 years. So I hope that you'll agree with me in the end, and in any case. So thinking about pattern recognition in run-on and sequencing data as a type of image recognition problem is really what gave rise to the DREG algorithm that we have used to identify enhancers and promoters in this kind of PRO-seq data. So if you take, for example, this region near the IL-6R gene, the PRO-seq tracks look like this, and then this is a summary of enhancers and promoters derived from, in this particular case, 38 different ChRO-seq experiments summarized using a tool called Chrome-HMM. So red and yellow determine the location of enhancers and promoters. And what we've done is we've basically taken the shape of the PRO-seq data, and we've used that shape to train a classifier called a support vector regression, which for each position in the genome, it gives a score. And that score, you can interpret a little bit like a ChRO-seq track. And you can see that it's high in these regions where we think there should be enhancers and promoters based on the histone modifications. So that's the way that DREG works. Using these same ideas, you might be able to predict where transcription factor binding is located, right? So if you take, for example, this region near the start of the KIF14 gene, DREG picks out this regulatory region right here near the promoter, and then there's 4 different ELF1 binding motifs within this region, 4 really good matches to the ELF1 binding motif. We would predict from first principles that it's this one which is in the regulatory region which is actually bound by ELF1, and indeed if we use ENCODE ChRO-seq data, we can confirm that that is true. So using this general strategy, Lauren Choate, who is a graduate student in my lab, was a previous graduate student in my lab, trained a classifier to predict the location of transcription factor binding genome-wide, essentially just by looking for this pattern that's associated with an active regulatory region. And her strategy achieved a mean area under the precision recall curve of between 0.69 and 0.72, which was state-of-the-art performance when she actually did this work in 2018. So in addition to that, more recently, Zhong Wang, who was a postdoc and programmer in my group, trained these kinds of models to predict the amount of different histone modifications. And he did this for histone modifications that fall into 3 groups, which we classify as active and punctate, which basically just means that there's high signal in a relatively short region, like an enhancer or promoter. And so, of course, this includes H3K27 acetylation, H3K4 trimethylation, which are 2 of the histone modifications that are used very routinely for mapping the different kinds of chromatin states within a particular cell type. Gene body histone marks like H3K36 trimethylation, as well as 2 repressive histone modifications. And I will say right now that the only one that doesn't work at all, that I'm going to spend minimal time on, is H3K9 trimethylation. So how does this process actually work? Does it work well or not? So to show you that, I've taken roughly a 2 megabase window on chromosome 21, which was a holdout. And I'm showing you here the PRO-seq data. And below that, I'm showing you ChRO-seq for H3K27 acetylation, as well as what we guess based on just this signature of transcription alone. And you can see that by and large, we're doing a reasonably good job picking out the location of peaks of H3K27 acetylation. We're not grossly overcalling, so there's not a whole lot of peaks that we're introducing that aren't actually there in the real data. So it's doing a decent job. And the same is true for all of these activating histone modifications, including those with slightly different patterns, like H3K for monomethylation is a little bit broader than most of these punctate marks. And then H3K36 trimethylation is high in gene bodies, and you can see we're doing a reasonably good job at that as well. And then to my surprise, we actually do a reasonably good job even for the repressive mark H3K27 trimethylation, where we're able to identify these boundaries of facultative heterochromatin along the genome. So it's doing all right there. So at more precise, higher resolution, if you zoom into one of the promoters within this window, you can see that we're capturing features such as the nucleosome-free region in this H3K4 trimethylation track, as well as the asymmetry between the transcribed and upstream divergent H3K4 trimethylation. And the same is true to a large extent for these other histone modifications as well. So when we're trying to understand how well the model is actually doing, I think it's important to put it in the context of what kind of accuracy do you expect by biological replication. So that's what I'm going to sort of walk you through on this particular slide. So here the x-axis represents the abundance of ChRO-seq signal for H3K4 trimethylation from Broad at all DNase I hypersensitive sites within K562 cells. And the y-axis is what we're guessing using this strategy called DHIT that Zhong has developed. So we're at a correlation of 0.78, which is not terrible. And it turns out that this is pretty close to what you get if you just compare ENCODE Broad to a different ChRO-seq data set produced by ENCODE USC. So we're sort of slightly behind biological replication. So if we generalize this not just to the ENCODE data sets, but to every public data set in K562 and GM12878 overall, that kind of information is summarized by these box and whiskers plots. So for each pair of data sets, we're computing a correlation, and then we're looking at the variation between all pairs of correlations in those two cell types for cell type match data. And what you see is that for these active marks, we're at about the median of any two biological replicates. So that leads us to suggest that for most of these active marks, we're sort of in the ballpark of where you expect biological replication to be, which indicates that transcription and these histone modifications share so much information that we do a really good job with just this single assay alone. So that's in two cell types. Does a model trained in K562 generalize to these additional cell types? Remember, if we're going to try to use this as a way to take a single assay and measure a patient sample, we want to be able to be sure that chromatin state in that sample is well reflected, right? So to address this question, what we did is we took our model trained in K562 cells and we looked at the correlations for all these histone marks. And then we used that model without any retraining to look at how good the correlation is in a variety of other cell types. And so we picked, of course, cancer cell lines like HCT116 and HeLa, where there's tons of data. But then we also have some primary cell types like CD4 positive T cells from human. We have liver of a horse, which changes both cell and tissue type and species. And we also have mouse embryonic stem cells. And what we find is that by and large, all of the active marks tend to have the same ballpark of correlation that we observe in K562 cells. And what this indicates, I think, is that the relationship between transcription and these histone modifications is a fundamental mechanistic property that doesn't vary with different trans environments, right? So we're able to take a model that we train in one cell type, and we're able to, by and large, expect that that model is going to translate into a new cell type that we can collect PRO-seq data for. So that's great. I really love the quote. I forget who said it, so let me know after if you know. But somebody has said, one of the best things that a model can do is fail, right? And the idea is that we can learn a ton of biology from cases where we get a prediction wrong. And so I became very interested in that. So we looked for cases for a while like this one here near I believe this is the ChRO-seq promoter, where we predict a ton of H3K27 acetylation because there's active transcription of the ChRO-seq gene. But the ENCODE data shows that there's just no transcription at all, despite having robust DNase I hypersensitivity. So a couple of things could be happening here, right? So the first is that there could be H3K27 acetylation could be dispensable at this position, right? So maybe there's some fundamental difference in the underlying biology where there's just no H3K27 acetylation here. The other possibility is that our K562 stocks are slightly different from the ENCODE K562 stocks, right? They're different genetically, they're different, they're grown with different hands in different FBS, and that culture media is slightly different. And so this may reflect fundamental differences in the cells themselves. So before I got too excited to distinguish between those, we did the ChRO-seq experiment in our own cell stocks. And so we didn't sequence nearly as deeply as ENCODE. But despite that, I think you can appreciate here that there's very clearly H3K27 acetylation at the ChRO-seq promoter in our data set. And so what this suggests to me is that the difference here at ChRO-seq and almost all places, there are a few hundred around the genome where this sort of complete on and off was true, almost all of them were explained by this. And it suggests to me that it's really fundamental differences between the K562 clones that we're growing, rather than differences between transcription and histone modifications, right? And so this really suggests to me that we see nearly the same variation in ChRO-seq, between ChRO-seq and transcription, as we expect between ChRO-seq biological replicates. Yeah. There's some really, it's sort of an aside for this particular talk, but there's some very fundamental biology that we've learned from this as well. And so one of these is relating to what the histone modifications, what role these histone modifications actually play in the in transcription by these cells. So this screenshot is from a 1964 paper in which Alfred Faulkner and Mernsky are the first, I believe, to report acetylation of core histones. They're the first to report that particular post-translational modification here. And in one of these great "it has not escaped our notice" statements, they speculate that maybe these histone modifications regulate gene expression, right? And this has sort of become something that the field really buys into over the last 70 years. But to me, the term regulate implies a causal relationship between histone modifications and transcription that is perhaps independent of the underlying DNA sequence, right? For them to be causal, they have to be able on their own to initiate this process. And is that really supported by the data, right? So this is still the subject of a lot of discussion, and an alternative perspective is that perhaps these histone modifications aren't themselves causal. Perhaps they serve as gears within this large transcriptional machinery apparatus. So that's sort of the alternative model, right? And I would argue that this very strong correlation between histone modifications and transcription already eliminates some of like the stronger models of the histone code hypothesis. But can we dig deeper to really understand this more cohesively? So Alex Shivu, who is a joint graduate student between my lab and John's, dug into this problem. And she asked whether we can learn something about the causal relationship between histone modifications and transcription, right? So the Alfred, Mernsky, and Faulkner view might draw an arrow in this direction, where histone modifications are causal to RNA polymerase II, right? And hence the information flows in this direction. Alternatively, if the COG model is true, there might also be an arrow in this direction where Pol2 is also necessary in some way for these histone modifications, right? So if this is really a large, complicated machine, if you take out part of the machine, perhaps the rest of the machine falls apart, in essence, is the hypothesis here. So Alex asked this question by blocking new transcription initiation by RNA polymerase using the small molecule triptylide, which targets the helicase TF2H. So in the presence of triptylide, the amount of transcription on genes decreases really rapidly. So this is PRO-seq data showing that by 1 and then 4 hours, you're decreasing the amount of transcription near transcription start sites almost to background. And the same is true as RNA polymerases that are already engaged run off the body of the transcription unit, okay? So what's remarkable is that when she does ChRO-seq for 2 different histone modifications that we think of as canonical promoter and enhancer modifications, H3K4-methyl-3 and H3K27 acetylation, she sees an almost immediate drop of the histone modification nearly to background by ChRO-seq, and then maybe it increases a little bit by 4 hours, but not up to the same level as it was before the experiment. And this happens at virtually all promoter and enhancer regions everywhere across the genome. And she can recapitulate this result by taking the chromatin-bound histones with each of these marks and doing a global ChIP Western, not ChIP Western, just chromatin-bound Western for these particular marks, right? And so this really supports this sort of bidirectional causality structure where transcription and histone marks are really interdependent on one another. And I would argue that this really much more strongly supports this gears model, this single molecular machine that then falls apart when you remove either piece of it, than it does a model where there's a causal impact of these histone modifications. So I'm going to switch gears now and tell you about how we can measure these things in individual cells, right? So we would like to be able to extract information about transcription and histone marks, these regulatory level and the gene expression level, in these individual cell populations. And so ideally, we would have a single cell PRO-seq assay. How's that going, John? That would be great. I mean, my lab will use this a lot once it's actually there, right? But it's a hard problem. And we'd like to be able to use the tools that we have and the data sets that we already have while that is being developed. And in addition to that, I'm going to argue that there's some real benefits of these bulk assays. So for single cell data, It's great that it provides information about single cells, but there's a bunch of biases that are associated with dissociation of the cells. So think about trying to cram this humongous neuron into this little droplet, right? It's just not going to happen, and this creates a bunch of biases. Plus, they're high cost, and they're not really scalable to the huge cohorts that we heard about in the last talk, right? And then there's kind of crappy data in each individual cell type. And the extent to which this matters, I guess we can debate. But by the same token, bulk assays are really scalable and well-implemented tools that work beautifully right now. And then there's thousands of existing data sets like the Cancer Genome Atlas for cancer that was discussed in the last talk as well. And really the main drawback is that there's no single cell information. So just to cut to the chase, I'm going to propose an alternative experimental design for something like this, where people, let's say that we're analyzing disease X, right? And we're analyzing glioblastoma or breast cancer or whatever it is. We'd want to collect the following data set. A limited amount of single cell information, just for the sake of argument, I'm going to say 3 to 10 individuals, but then large numbers of bulk samples, hundreds, thousands, whatever we can get the funder to pay for. That's what we collect, right? And I'm going to argue that by using the right computational tools to integrate these two data sets, we can drill down and get the best of both worlds, okay? So our strategy to do this was really the brainchild of Tianyi Chu, who was one of the best graduate students I've ever worked with, and I was so lucky to recruit him as my first graduate student. He developed this strategy right before he left the lab called Bayes Prism. And Bayes Prism is essentially a way to predict both cell types and the gene expression, the average gene expression in each cell type from a bulk RNA-seq sample using a single cell RNA-seq sample. Okay, it's a Bayesian inference strategy that essentially, like a prism, takes this vector of counts representing the gene expression of individual genes in a bulk sample and splits it into a matrix, where each row in this matrix represents the vector of counts from each cell in your single cell data, each cell type in your single cell data, okay? So it's a Bayesian strategy. I won't go much into the underlying math or computation, but Tianyi has I think it's like a 20-page supplementary note 1 in his paper. This was published a few weeks ago, that goes over math, all of the math that actually underpins this strategy. It's a beautiful piece of work. So check that out if you're interested, and get in touch with Tianyi or I. For our needs, the question is, does it work to infer single cell kinds of information? So there were a bunch of benchmarks in Tianyi's paper. The one that I find most convincing is 2 data sets in glioblastoma that were done by different labs using different technologies at different times, okay? And so there's a lot of technical variation because there's 2 different platforms where the samples were run on. And then there's a lot of biological variation because no 2 patients will have quite the same gene expression pattern in their tumor cells, okay? And despite that, Bayes Prism is able to infer, so this y-axis is what Tianyi predicts using Bayes Prism as the cell fraction for these cell types that are represented in the single cell RNA-seq data. And then the x-axis is the true fraction that we know because this is fundamentally a single cell RNA-seq data that Tianyi threw away the cell type labels for. So this works pretty well for deconvolution, right? In addition to that, Tianyi can also predict gene expression in each one of these cell types that he's looking at, okay? So how does this work? So as I'm showing you the data, I'm going to remind you that due to cross-patient heterogeneity, having information about gene expression in one of these tumors gives very little information about gene expression in another tumor. And so that's why when you see this UMAP, so this is 28 patients, each one, I'm only showing malignant cells, and each patient has a separate color here, right? So that cross-patient heterogeneity is represented by this patchiness, right? So each patient is a slightly different gene expression profile, and that's why they form these patches in the UMAP space. So for most of these, when Tianyi predicts gene expression by Bayes Prism, that inferred value, which is represented by the triangle of color in this UMAP here, is just about right over where the mean of expression of all of these single cells is for that particular patient. So he's doing a pretty good job in this task too, I would argue. So this is a really powerful strategy. And just to illustrate one of the kinds of questions that we could answer with this particular tool that we couldn't answer with previous versions of deconvolution strategies, I'll show you just two very quick slides on one of the vignettes that Tigny told in his paper. So Tigny took data from the Cancer Genome Atlas, and he focused on a little over 1,000 samples in 3 tumor types. So this includes glioblastoma, head and neck squamous cell carcinoma, and melanoma. And he deconvolved them, right? Because there's so many samples here, he can predict the correlations between clinical outcomes and the presence or absence, the amount of each cell type within each tumor. And one of these that was a particularly interesting story was macrophages, or in glioblastoma, it's probably microglia mostly, but I will say macrophage, so forgive me for that if you're an immunologist. In melanoma, there was a very strong correlation where macrophages tended to be very pro-survival. So patients that had a lot of macrophages tended to live for longer despite having metastatic melanoma, which is a really nasty type of cancer. In head and neck cancer, there wasn't really much correlation with macrophages. And in glioblastoma, high amounts of macrophages was worse for prognosis. So what is going on here, right? Is this specific intricacies of each one of these different tumor types? Is it something about the macrophages or the microenvironment of the tumor? And can we work that out? So it turns out that there's really 2 different kinds of macrophage or microglia, M1 and M2, and they have different roles. So M1 macrophages tend to be much more immunogenic. They mount a strong immune response, and they are thought to kill tumors, whereas M2 are much more associated with wound healing, and you might expect them to fix the tumor, I think is the idea. So Tigny dove into the expression estimates from Bayes' prism for each one of these bulk samples, okay? And that's what I'm showing you here in the scatter plot. So the M1 macrophage similarity for each bulk sample is shown by the position on the x-axis, and the M2 similarity is shown by the position in the y-axis. And each one of the dots represents one of the bulk samples that Tigny is estimating expression in. And what you can see by this is that macrophage polarization completely explains the differences between these different tumor types, right? So glioblastoma, shown here in pink, tend to have very high M2 scores, which is associated with this much more tumor permissive role. Whereas in melanoma, macrophages tend to be much more M1-like, which is associated with a much more immunogenic response. And in addition to that, if we just take one of the tumor types, in this case I'm showing data from melanoma, but the same was roughly true in glioblastoma, the macrophage polarization based on gene expression estimated by Bayes' prism was even more informative about the duration of patient survival in Tigny's Kaplan-Meier plot analysis. So this really is something that we would have completely missed without that extra dimension of expression information and without having large cohorts to be able to investigate, okay? So I think this really showcases the benefits of this particular kind of a strategy. So next we'd like to be able to take this kind of a tool and deconvolve bulk PRO-seq data using either single-cell RNA-seq or single-cell attack-seq. This plot here shows the correlation between DNase I hypersensitivity as a measure for chromatin accessibility and what Zhang is able to predict using his d-hit algorithm. And what it shows is that for a subset of sites, there is a very strong correlation between transcription and chromatin accessibility. And it's this correlation that we're going to use to take single-cell attack-seq as our prior and deconvolve bulk PRO-seq data, okay? This is still an active research project. It's led by Blaine Harlan, who is now a postdoc in my lab and previously did her PhD with Paul Soloway. And she is fantastic at both the bench and at these bioinformatic tools. And what she's gotten done so far, since she joined the lab in January, is she's generated single-cell attack-seq data from 3 primary human glioblastomas, as well as matched PRO-seq data from exactly the same nuclei preps, okay? I7-1, I7-2, and I7-3. And she can use these to get a look at what chromatin accessibility looks like in glioblastoma. But in addition to that, she can also use this to ask how accurately she's able to develop the tools to deconvolve the bulk ChRO-seq data using these chromatin accessibility patterns. So here she's using a leave one out study design where she takes I7.1 and deconvolves it from the PRO-seq data using I7.2 and I7.3. And it works fairly well. The latest version, I should say, of her strategy works fairly well. So here I'm showing you 2 of the 3 tumors. It works similarly in the third one. And she can deconvolve these with fairly high accuracy using leave one out. And then in addition to that, she's also able to predict chromatin accessibility in each one of these cell subpopulations, the tumor or the immune subpopulation, with a fairly high correlation to the ground truth measured by single-cell ATAC-seq in this case. So I'm really excited about this because I think it's really a great window into disease that we don't really have right now, okay? To really showcase this to the community, we've combined Blaine's 3 single-cell ATAC-seq data sets that she's collected. I should mention this is a collaboration with Paul Soloway as well, where Blaine actually did that data collection work. And we have data, PRO-seq data from 102 different primary glioblastomas at this point. And we can use these patterns to generate information about single cell and be able to deconvolve these. Here I'm showing the first 69 that were sequenced and mapped to the genome. So for each one of these patients, we have each of the cell types, and we have the chromatin accessibility in each of those cells, estimated with fairly high accuracy. So that is, I think, going to be a tremendous resource to really understanding this particular tumor type. So just to summarize this most of the talk, PRO-seq is a great assay. It's an assay that maximizes the information that you gain from a particular sample using a single experiment. And I'm going to argue that due to these, thanks to that property, as well as some of these bioinformatic tools, we're able to use a PRO-seq data set to ask the kinds of questions that may have only been the domain of large consortia. But we can bring the cost down where you could do it in your lab for the cost of an R01. I think that's really the key take-home goal of this technology development. In the last 3 minutes, I'm going to describe a quick vignette of why we need this kind of regulatory information in the first place. What can we learn that we can't learn with just gene expression? And so to do that, it's instructive for me to take you through a hypothesis of how this whole process works, right? So we start with transcription factors, and transcription factors, just their biological function is to regulate groups of genes, and these target genes carry out a particular biological function at the cellular level, right? So either stemness or immune infiltration or that kind of thing is what I'll show you. And these biological functions cause changes in clinical outcomes. And I know this is not news to anybody, right? I'm not teaching you anything here. This is the way that we all think about it. But the reason that I'm showing this is because it really highlights this idea that maybe transcription factors serve as a better biomarker in a lot of ways than the individual genes which are within the programs that they regulate. Okay? So that's sort of where I'm going with this whole procedure. So Tini took the first 20 glioblastomas that we had ChRO-seq data for, and he conducted an analysis where he looked at which regions are controlling gene expression with PRO-seq. And he wanted to get to the biological functions that these different regulatory regions were driving. So in order to do that, he's using a strategy where he's looking at guilt by association by intersecting those with a large compendium of DNase I hypersensitive sites from lots of different tissues collected by Roadmap Epigenomics Consortium. Okay? And so for a particular tumor in our cohort, if you do that sort of intersection, what you find is that for about 6,200 of the regulatory elements that are active in that tumor, they're active in every single tissue in the body. And of course, these are constitutively active promoter regions. Okay? But the other really interesting thing is that there's sort of another peak of this distribution out here. So these are regulatory elements that are active in a couple of tissues, but really, really rarely. And Tini had the insight that which tissues were active give you information about the biological function that you're actually looking for. Okay? So that's what he's looking for. And not surprisingly, the majority of regulatory elements in each of these samples intersected with normal brain. And of course, this is a tumor of the central nervous system, so we're not surprised about that. And there's a few that are tumor-associated, but not normal adult human brain. So what are those? Tini devised a sampling strategy to assess the surprise, the degree of surprise with which so how strong is the match between a particular tumor in each one of these tissues. And you'll see that in some cases, like GBM 1590 here, there was very strong enrichment relative to this null distribution in the fetal brain. So this is consistent with the idea of glioblastoma as a dedifferentiation to a more stem-like state. But that wasn't true for all of the glioblastomas in the cohort. So GBM 8804 had a very different signature that was much less stem-like. So he took all of the tissues and he clustered them into 3 different regulatory programs that he argued contained insight into the cellular functions that these things were driving. And these 3 are shown here. So fetal brain is in the stem-like program, which was very prevalent in the glioblastomas. There's also an immune program and a differentiated program, which contains cells like astrocytes and fibroblasts that maybe form a supportive role in a particular tissue. And he noticed that these 3 programs were active to different degrees in different tumors. Like, for example, GBM1 was very high in this fetal and stem-like program, whereas GBM2 was much stronger in this immune program. Okay? And then he identified transcription factors in each one of these regulatory programs. So because we have weak power to differentiate between different paralogs, right? Like we can't differentiate between 2 isoforms or paralogs of like nanog, Tini clustered these specificities, and that's what's sort of shown here. So here, all of the rows here, and I'm sorry that this pointer works so badly to actually point, represent AP1-like binding motifs, which are really enriched in the differentiated program. And in cases where we know something about this, like this POU domain containing, this is nanog in essence, and some of the genes that encode nanog are required for tumor initiation in models of mice for this particular cancer. So this sort of jibes with what we know about the tumor. And then Tini asked which of these transcription factors were associated with a clinical level covariate, in this case, a clinical outcome. And he found 3 that were correlated with clinical outcomes: CEBP, REL-A, or you might know it as NF-kappa B, and RAR gamma, so a retinoic acid receptor that was described in one of the talks this morning. And of these 3, only CEBP was known to be associated with clinical outcomes, because if you look at the abundance of mRNA for CEBP, that correlates with survival just as the activity of the transcription factor does. But for the other 2, that wasn't true at all, because, I mean, a great example is retinoic acid receptor. It requires that extrinsic retinoic acid event. It requires an external event to activate that transcription factor and cause it to have its effect on chromatin. So I think this really showcases how we can get something out of transcription factor function that we can't necessarily get out of just gene expression alone. So I'll stop there. What I want you to take away from this is that PRO-seq and the variant ChRO-seq, which we can use in tissue samples, is a great strategy that maximizes the information about what the genome is doing in a particular sample. And then there's tons of applications for these things in your disease of choice. And you too can use these tools and get in touch with me if you want to chat. So thanks to everyone.
Can open it up for questions.
Wow, amazing talk and fantastic tools, Charles.
Thanks, Haiwan.
So I'm curious about using base pair prism to deconvolute PRO-seq data. As you know, we talked before, we looked into deconvolute PRO-cap. I'm wondering, it seems like it works really well, but how does it work for the enhancer regions? Does it work well? Yeah, because we found the problem is that they're often in 2 different cell types. They're both open, but the PRO-cap signal only are in one cell type, not in the other. Those are the hardest for the lead count.
Yeah, so this suddenly got very loud. So you certainly, you can have chromatin accessibility without having transcription. Absolutely true. And I mean, I think you have to be a little bit careful as you interpret these. You don't necessarily want to say that this accessible region is transcribed in this cell type. I think what you're sort of doing, the way that I had this discussion with Blaine that I think really clarified things is think about taking the transcription signal, imputing chromatin accessibility, right? So you have predicted chromatin accessibility, and that's going to have a little bit of error in it. Arguably, I mean, I would argue it's not that much error in the grand scheme of things, but it is going to have a little bit of an error. And so what you're predicting isn't necessarily transcription. It's that imputed guesstimate of chromatin accessibility, and that seems to correlate pretty well with overall chromatin accessibility, at least enough to do the kinds of analyses that I think we would want to do in a lot of tumors. But you're absolutely right. I wouldn't necessarily be comfortable classifying something as transcribed or not transcribed in cell type X. Yeah, it's really just that correlation with imputed chromatin accessibility. Yeah.
Yeah, so really interesting thinking about deconvolution and your macrophages. The tool and the database that you used, are you thinking about interaction between treatment and prognosis, or it doesn't really matter?
We would love, yeah, I mean, there's all sorts of questions you can ask with this tool, I think is the answer. And you can ask about how treatment affects a particular cell type composition and in turn gene expression. You can definitely design an experiment to set that up and use this tool to do that. You can ask about how it certainly probably has prognostic implications as well, or prognostic applications as well, that it would be really great to explore.
And then my follow-up question. So these are very expensive experiments to perform. Right?
They are for the single cell data, right? It's maybe, I mean, what would you say? Maybe if you only use 2 biotin, so if you take some shortcuts that decrease the cost but don't necessarily affect the interpretation of the data for cancer genomics, you could do these for $50 an experiment, as little as that if you really got it scaled up. So I think that you really could do hundreds of patients in an R01 sized grant.
Because we're running a little behind schedule, I'd encourage others with questions to catch up with Charles outside. We're going to take a 15-minute coffee break, roughly, and come back at 3:45. Thanks, Charles, for the drip talk.
Thanks, everybody.